delay. There's a little bit of a delay between when it starts here and what you see there.
Okay.
So, but you need to turn the sound off because otherwise you're going to hear me over there and over here. Hopefully. Am I showing up? Okay. Just making sure you got it set up. So today we're doing something a little different. We are streaming from my Stereo Microscope in the actual light microscope lab. So I also have other light microscope, compound microscopes in the lab. But for today, we're going to be using this one, which is pretty different than the types of microscopes that we normally use. So a stereo microscope, sometimes called a binocular microscope or a picking microscope or a dissecting microscope. It's basically like a pair of binoculars. So you have the eyepieces up here and then a mirror, and basically it just funnels it right down onto a lens. And so what you're seeing is I've got these fiber optic cables where I can kind of move them around and point them at different aspects of what we're looking at. And right now, I have a little piece of it's a bit of the lunar moth that Miss Daisy D sent to us that we put on the scanning electron microscope. And I thought it would be kind of fun to take a look at it in the light microscope, in part because in the scanning electron microscope, we don't see any color because there's no light, and we're mostly using, well, we're entirely using electrons in order to see. And in the light microscope, you can actually see, in the stereoscope, you can actually see the actual colors. So a lot of times for the scanning electron microscope images, I'll colorize the image. So I might be inspired a little bit by the ideas of things that I see in nature, but most of the time it's just sort of like things from my imagination. Is it a dead sheep? No. It's a tiny piece of a wing of a lunar moth. Hello, Pacific plankton. So like I've got a little bit of lunar moth wing left from when Miss Daisy D sent us some lunar moth material, and right now we're looking at it in the stereo microscope. So I know, I guess you think that looks a little bit like an eyeball or something, but you're actually seeing an eye spot on a lunar moth wing. So a tiny dragonfire, hello, and also Ribozoid, hello, and DJ Technique, how you doing? So I'm in the light microscope lab, so my settings are a little bit different, and then also not completely obvious to you is back here behind me at a desk that's sort of a safe distance from me, and also masked up is my PhD student Sukanya.
Daddy is cute, Daddy is strong, Daddy is brave, and Daddy will not stop.
A subscription from Pacific Plankton, thank you. 6 months, it's basically how long I've been on, the entire time I've been on Twitch. So thank you for that, Pacific Plankton. We've got a good thing going, sort of sharing our microscope stuff here on Twitch. Andalor says that that eye is really pretty convincing, and I would agree with him. So I'm going to manipulate it a little bit. The funny part is this stereo microscope has actually got a lot lower magnification power than any of the other microscopes that I have in the lab under different conditions. I actually have a 2x nose extender on this, so I've bought sort of the high-end equipment for it, and I've got 16x objectives on the eyepieces instead of 10x objectives. It means that we can actually get some pretty high quality, very high resolution images out of it. One of the things that's cool about a stereo microscope, though, is that kind of like the scanning electron microscope, I can change the magnification, and we are actually not the closest that we can be. We're not even nearly as close as we could be. So this is it was starting off in the most distant position, which was 0.55 times, and I'm just going to keep zooming in. Ignore the scale bar in the corner. It doesn't know what it's talking about because I'm changing the magnification wheel, and it's just geared towards the base magnification. So you could multiply that value times 2 right now, and it would probably be accurate. And what you can see is the same thing that we saw in the scanning electron microscope, which is that the moth scales in this case are actually these sort of like the things that we think of as being little hairs are actually little scales, and the wing is entirely covered by these structures. And I can still keep zooming in. We're not at the limit of my magnification, so this is 3 times the original image's size, and what's in the center of the field is not in focus because I'm kind of focused on the edge to get the colors that are here. One of the things that's a little more challenging is that all of my other microscopes have a sort of an X and a Y controller, whereas the stereo microscope, just you push things around on it. It doesn't have an easy way to control things.
So are these scales pigmented or is it color, structural color?
Yeah, so the question is it are there pigments in the scales or is it structural color? For these scales, I think they're actually pigmented because it doesn't matter which way I turn them. They still have more or less the same color. And there's definitely some in there that are this sort of really dark color and really light colors, and then the yellow and the sort of reddish pink color that's there that you can see really clearly. And I can zoom in even farther. We're still at 3x, so here's 4 times and here's 5 times. So we can actually get a really nice high resolution image out of this, and you can see quite a bit in the light microscope, or in the eyepieces rather. I'm actually focused here in this sort of dark part in the middle. So one of the things that happens when you are imaging things like this is that the topography means that very little of the actual image can be in focus at the same time. So when I zoom out, you can kind of see that there's blurred areas and there's areas that aren't blurred. So I could be focused way up here on these hairy things on the right end of the screen, or down here on the sort of wider scales. Those little hairs that are up there are actually scales as well. So if you were to look at them in the scanning electron microscope, or if we would be able to get just a little bit closer than we can with the stereo microscope, you'd actually be able to see that that they're actually the same kind of composition as these scales that we can see in the middle part of the screen that have all the bright colors on them. So what I'm going to show you is one of the cool things that this microscope is actually capable of doing, or rather the software associated with the microscope is capable of doing, and hopefully I can make it work, which is what we call a Z-build, or a live Z-build. And that is fine. I don't care what you call it. It just wants me to name stuff. And then what I can do is start that build, and as I focus through from one end of it to the other, it will build that image, and it will only put the things that are in focus in focus. So as I focus through the image, what happens is you'll see things that are on the right side that were completely out of focus in the live view, and things that are in the middle also in focus at the same time. And so I can create this image that looks like we're from much farther away. We would have the sort of regular depth of field, but in fact we're getting the magnification that we need across that sort of range of different Z values. So, and then when I'm done, I just click this button again. Hopefully it works. And it turns off the stacking. And then I could save that image, right? So I have a whole image that's created all at once with just by changing the focal height. So it's kind of cool because now I have the ability to see the whole thing in focus, which isn't real. I mean, in real life, it wouldn't look like that because of the Z-stacking tool. So we can kind of go back and you'll see that that image is not that way. So there's positives and negatives to this, of course. One negative is if you're imaging something like this and you actually do the image stacking, what happens is it looks kind of flat when it might be something with a lot of topography. So because we're getting rid of the depth of field aspect of it, basically, it removes that sort of topographic measure from what we actually see. Hey, Del Maxim, hello, and Sarah, hello, and Rihanna, hello. Let's see, what did we have? It's a pretty nice view of just the scale part of the moth. One of the things I would say is that the color is a little bit adjusted a little bit wrong. From what I can see in the light microscope, the yellows are just a little bit off, and I could use some sort of white balancing technique to get the color to be a little bit more like it looks in reality. But for now, I'll just leave it as it is. So this is a pretty cool tool. It's a little bit different. It gives us sort of the middle scale magnification that we might be really interested in seeing. We could capture the same image in a scanning electron microscope. It would look basically the same except for black and white, but the resolution of the scales would be a lot better. So we'd actually be able to see the little tiny holes and things basically that the scales have in them. I should point out that I made some new chat commands. So if we're using the stereo microscope and you want to provide somebody with a little help, what's the stereo microscope do? There's the actual stereo microscope that we use. This one has an integrated high definition camera. So the eyepiece and the magnifier and the everything that's on here is basically has a built-in camera that's it's part of the actual instrument. And I actually have, when the nose extender isn't on, I can use this set of ring lights that can be attached to the front end of it. And the ring lights are basically just an LED light system. But it provides a lot more light, basically straight up and down. And sometimes you want that. Sometimes you don't. For some things, you might want to be able to reposition the light. If you wanted to try to sort of see shadows or whatever, do some puppeteering if you're mind of a snail, you would do this sort of thing where you can basically change where the light is. And it's kind of nice because it's not a static light position, so it's more like what happens when you move objects around in a room or something. So I actually kind of like that as a capacitor, as a tool. So this is a Luna moth wing. And then also I wanted to mention a couple of the commands that I also added in here. So one of them is this one, in case you wanted to see when Pacific plankton streams, or if Dell's in here and you want to give Dell a shout out, you can just now shorthand those ones. So regular people with regular hours that I can mention to people. So as I said, it's sometimes easier for me to zoom out so you can kind of get a sense of what's going on, but also so I can figure out how to move what I'm looking at, because I'm just pushing it around with my fingers, which is kind of challenging. And so that's the head of the moth, just to give you a little bit of a sense of that. And I think I can get that centered. And then we could go look at the antenna a little bit. So because of the really big differences in height, it's sometimes challenging also to get things focused the way that you want. But you can see in from this, in the middle of that image, that the little hairs that are sticking off of the moth's antenna, not just the sort of like dendritic or tree-like shape to them, but the actual little tiny hairs that are on there. And in the scanning electron microscope, we were able to see those hairs pretty clearly, and they stood out pretty strongly. And there was sort of like a scaly covering over the surface of these things, and you can get a good sense of those little hairs. Each one of those is basically it's like how it smells, the environment, how it finds what it's looking for out there. Usually, it's a mate. So this is a pretty nice view of just the little antennas of a Luna moth, and we can get a nice close-up of those. So that's the maximum magnification that I can reach with the setup the way that it is right now. But you can still see those little hairs pretty clearly at this magnification. So yeah, chemoreceptors. So for this, I don't actually study Luna moths. This is just some materials that we had on the scanning electron microscope that Miss Daisy D sent me, and she wanted to have us check them out in the scanning electron microscope, and I thought that would be amazing, so I agreed to that. And of course it was a little while ago. I think we did those back in the fall. And I've had it on the SEM a couple of times for streams, but we've never looked at anything like this. We've never used the Stereoscope before for streaming, so I had to do a little bit of futzing around to get the Stereoscope working correctly with everything that we usually have, like the camera so that you can see me, and actually gives me the ability, if I wanted to kind of move this, right, because it's just a little webcam that I can show things around, and can point back there so you can see Sukanya hanging out on the desk in the background behind me with a mask on, being safe, of course. And I can kind of reposition this a little bit so you can see a bit of what I'm doing. So that's the antenna. Let me move this around a little bit because I don't know if you noticed there's the head of a moth not too far off of the screen. There's its actual face. Now the lights aren't perfect for that, so I'll see if I can fix that. The extension for the 2x extension actually kind of blocks. So one of the reasons it's a little bit troublesome is it kind of blocks the light. There's only sort of a limited amount of working space capable for it, and so that sometimes creates a bit of a problem. But now I can zoom in on just the eyeball of the moth. It's a little out of focus, of course, but I can fix that. So you can see the little tiny compound eye components on the moth's eye, and you can also see this is a dead moth, of course, and you can see that this one's eye has a hole that got poked in it somehow. It just got fractured, basically, and so we can get a nice close-up view of that compound eye. We're not done zooming in, of course. That's the maximum right there. So I think I did some calculations, and when I'm completely zoomed in, we're still not all the way zoomed in, by the way. There's got a little bit farther we can go. At this level, we're probably around, I think when I calculated it with all of the eyepiece and the 2x extenders and everything else, that this view is about 380 times magnification or something like that. So equivalent to sort of a 40x on a light microscope, which is really pretty good for a stereo microscope. Not the sort of level that I was kind of expecting. But what that means is that we could potentially look at all kinds of things that are very small. So that puts us in the range of being able to see many of the microscopic aquatic organisms that I look at. So for my actual research. So this is just the eye with its little fracture in it. And if I zoom out, of course, you can kind of get a sense of how close we were. So we're just looking at really the head of a little moth. And if you need some other aspects to get a sense of the scale,
What's the magnification?
it's almost 400 times. That's my fingernail. right? So just a little bit of it that you're seeing is my my fingernail. So I can focus on my fingernail if you like, if I can hold it still enough, right? So just give you a sense of having a relative scale. That's my fingernail in focus, right? I keep my fingerprints away from the screen so you can't dox me by my fingerprints. But it'll give you a pretty good sense of roughly how small those things are. Fingers aren't always a standard size, but I don't have a lot of other things that are a standard size that I could stick under there. That's a piece of a tweezer head, if that helps. If you know what forceps size is, basically that's the head of a tweezer, so very small. Very, very small in some cases. The reason I can't wear, or I opted not to wear a mask and have Sukanya sit really far back was when I wear the mask and I'm looking through the eyepieces, it fogs over the eyepieces and I can't see. So like, in order to make it so I can actually see what I'm doing in here, I needed to have the mask off. But Sukanya has her mask on and she's quite far away from me, so I feel like we're probably safe. Oh yeah, a pencil would probably be good. I don't have one of those over here. But just to give you a sense of like how zoomed in we could be or are, right? It's not a dead rabbit. It's the head of a Luna moth. Just a moth, a tiny little Luna moth. And you can see both eyes, kind of, if I focus right here. You can kind of see both eyes of their heads. They don't really have a neck, right? They just have like their body and then the eyes sticking off of it. And I can move it around a little bit if you want to sort of get a sense of, they're fuzzy, right? So you can kind of see how fuzzy they are. Let me zoom out. Oh, that's as far out as we can go, unfortunately. So we're at the outward limit of it. But you can get a sense of this sort of, they're kind of a light green colored moth with little eye spots on them. And so you can kind of see the eye spots and the colors associated with the scales that they have. So like here, that's my fingernail. This is, if it were a sheep or a rabbit, my fingers would be enormous. So you can kind of see right on the front edge of their wings, they have like a red and an orange color that's really distinct for the Luna moths. And, we can actually get in real close to that, right?
What does the fur do for its aerodynamics?
That's a good question. What does the sort of fur do? Is that what you asked?
Yeah, what does the fur do for its aerodynamics?
It probably helps it stay aloft. I don't know if you've ever watched a moth or a butterfly when they fly, but they kind of do this, right? So they probably need a lot of resistance in order to get a lot of traction for their wings. There's some studies that have shown that the scales might also help them hide from bats, because the bats use echolocation and they eat moths. So there's some studies that suggest that maybe their tail wings are sort of shaped in a way that allows them to, be sort of semi-invisible to moths. But I don't know. I'm not a person who studies, or being invisible to bats rather. But I don't know. I'm not somebody who studies moths for a living. But I think it's kind of cool to be able to look at them because they are one of these sort of really charismatic organisms that, at this scale, at this sort of middle scale. So I thought I would start with that. I wasn't going to spend the whole day looking at moths. So I have a bunch of other things that I thought I could stick on here on the Stereoscope that we've looked at in either the light microscope or the SEM before, to try to give people some sense of the middle scale, right? This sort of middle zone that we're in for this stream. So this moth is actually, it is not, the body's missing from it. So for clarity, here's what we were looking at under the machine, the instrument, and So you've probably seen these types of moths before, but its body's missing, right? That's the way it was sent to me from Daisy. But you can kind of get a good sense of both the color and the sort of eye spot structures, and there's its head, right? So really pretty type of moth. And I have one that's not been destroyed in there as well, that we keep around in case we need to take a look at something like that. So I'm going to put something else on the stereoscope now. So it's a lot easier to replace my materials than normally, because in the SEM, when I'm streaming from the SEM, we have everything in a vacuum, and now I can just stick my hand in there and move stuff around. So another thing that I think looks really nice in these sort of scales is lichen. So I grabbed some lichen off of a tree and brought that in. Again, we've looked at lichen quite a bit in the scanning electron microscope. This is the real color for this lichen. It's a little zoomed in, so you probably can't get a good understanding of what we're doing. But so that's the bark is the dark brown that you're seeing, or the light brown that you're seeing in various places, and then the actual lichen that you're seeing is a bright yellow color, and that's the actual color of the lichen. Actually, in some parts, it looks kind of reddish or orange, and that's actually like real color for the lichen. Again, those aren't. That's not an illusion. There's nothing wrong with the color for these. It's maybe a little bit, a little less green than it needs to be, but other than that, those are real colors. So in real life, if you were to see this with your eye.
Good news, everyone!
Another follow, thank you, from Blade Runner. You can kind of see, can give you a sense of it, right? It just looks like a little bit of yellow on the outside of the bark. And to give you some idea of scale, we would never be able to see that it was actually little tiny blades like it looks right there, without a magnifying glass of some type, probably. So stereoscopes are basically just fancy magnifying glasses where you have both eyes available. What's the question, Sukanya?
Yeah. How does the moss transport nutrients? Lichens.
Oh, how does the lichen transport?
Yeah.
Lichen are kind of interesting organisms. They are a combination of two different organisms normally, and so they do a little bit of housing for an algae that is photosynthetic, and then there's also some bacteria, and I think the community of a lichen actually, so some of it's autotrophic, and some of it they get some nutrients from the surrounding environment, but so they might pick particular trees that they want to grow on, but you can also see lichens growing on sidewalks, right? So they don't necessarily need to get a lot of nutrients from their surrounding environment. They can get a fair amount of them just from the endosymbiotic relationship they have with the algae.
Does it have vasculature?
Does it have? Oh, no. So lichen don't have like a root or a vascular system at all. And some of these we might actually be able to see. It's not a plant. It's a mixture between, as I mentioned, algae and a fungi, I think. Somebody out there must know. And then also a bacteria. So it's like three organisms that are grouped together, and they basically function as one organism. But it's not a plant at all. So it doesn't have a vascular system like plants do that funnel material from a root up to a leaf or anything like that. This is the structure. The whole organism is here, and it's actually a combination of three organisms or more sometimes. I think there's also a yeast component sometimes, if you can believe that.
Is lichen seen as a pest?
No. They're too small to really be a pest. I mean, they grow on sidewalks and trees and stuff, but they're totally commensal. They help themselves, but they don't hurt the tree really in any way. They're growing on the bark. They're not growing on the leaves. If anything, they might function to protect, the trunk from damage a little bit, from either UV radiation or weather conditions. For rocks, they're kind of interesting, the ones that grow on rocks, because they can actually kind of create some sort of acids that will chelate metals and things like that. And actually, they can get some nutrients from rocks, but the normal process is like they sort of weather the rock directly under them for a little bit and consume the nutrients, and then it actually typically acts to protect the rock from weathering. So if you have a surface of a rock that gets covered with lichen, like over a really long time, the lichen probably protects the rock more than it hurts it, like on balance. Initially, it probably creates problems for the surface of the rock, but I mean, we don't really care about rock surfaces, do we? Other than just making it a little different color, it doesn't really have any impact. And I think that a lot of the lichens are actually really pretty colors, right? So they make bright greens and yellows and reds and oranges. So for most rocks, that's an improvement. They go from like an ugly brown or gray to having all these sort of bright golden colors.
Is a fungus considered a plant?
No, fungus is not a plant. A fungus is actually closer to a human than it is to a plant, believe it or not. They don't belong in that group at all. Funguses typically are mineralizers. They're decomposers. They're on the other end of the sort of circle for life. And plants are autotrophs. They're on the sort of producer side of things. And the reason that this thing has a symbiotic relationship with an algae is that the two can live together. So typically the fungus will provide some sort of structure and sort of safety for or housing for the algae, and then the algae then generates food. So it's a relationship that's from the way that the organisms grow, that they are probably somewhat inseparable now. Like if you took them apart, both organisms would probably die. So
How does no transport nutrients to grow?
how does
They're saying like mosses are non-vascular plant.
well, these aren't mosses. Actually, I have some mosses we can look at. But I'm not an expert in mosses either. But the bryophytes are kind of interesting group of mosses. I don't think that all the mosses function
Good news, everyone!
exactly the same. Thank you for the follow. I really need to turn that volume off. I thought I had the sound completely off on this thing, but it keeps playing sounds anyway. So what can I do? Anyway, so this is sort of a cool a view, I think, of lichen, and of course we're not zoomed all the way in, so you can actually get right up in there and see the structure of the lichen pretty clearly. And then it's got like a sort of a sparkly, glittery surface to it. That's sort of like the hard crust of the lichen itself. And then we can actually see the structure of the bark a little bit there. It's got some dust trapped in it. So
Good news, everyone!
Still see the dust as well. I wish the speakers would listen to what I was telling them to do, but they don't. It just keeps yelling, "Good news, everyone!" But that's fine. We'll take the good news. Thank you for the follow, despite the fact that it keeps playing when I tell it not to play in my ear. Thank you. So, yeah, we can get a really nice clean view of some of this stuff, get zoomed in on it, and here you go. You can kind of see really clearly the kind of, at the ends of it, little buds that might then ultimately grow into this sort of broader surfaces that make up most of the lichen that we were seeing. So you can see that really clearly there. There's little sort of fuzzy bumps on the ends of the lichen. So again, we saw a lot of this stuff in this same sort of stuff in the scanning electron microscope. You could see them sort of like branched structure that they have.
How do lichen reproduce if they are almost separate organisms?
I don't know the answer to that. I'm sorry. I had to look it up to be able to answer that question. I don't actually know how they reproduce. Probably they just get regrow from fragments. They might actually, it's a fungus, so they might have spores that, they might have a season where they actually go through a sporing process. That's the way fungus typically reproduce. But I'm not an expert in lichen, so I can't answer that question. I maybe should find an expert in lichen so that we could have them on the stream and I could talk to them a little bit about it. Let's actually. So you're asking about mosses. I can put on a moss. Maybe OpenSet would know. He seems to know a lot about these kinds of things. So OpenSet shows up. Maybe he can answer some of those questions. So I'm actually just playing with stuff on the microscope and making it somewhat educational, I hope. So this little thing that we're looking at here is a moss. And again, this is what I plucked off of a tree. So on the back side is bark. And on the front side is the actual moss. And these might be found on any local tree around your where you live. But you can kind of see that that this is definitely different than the lichen. There's little tiny sort of pieces coming off of it, right, that are like strands. So this is an actual moss. And probably if I wet this, there can be sometimes little things will sort of start moving around in it. So a lot of times mosses like this have little springtails hiding in them. So if you put some water on it, you might get some springtails that wake up and start moving around. They're sort of an important ecosystem for these things that live in and among them. The movement that you're seeing there is actually just a piece of dirt that's trapped in the other than when I'm moving my hand. A little piece of dirt that you can see trapped in them. But again, there's the surface of one of these mosses. So, like before, we can zoom in like crazy close, and we'd actually see the cellular structure, believe it or not. So I can see if I can get real close for that. These aren't the sorts of things that we could actually see in, I can put water samples and look at in the light microscope because they're semi-transparent, whereas for the most part, the lichens are too opaque to see through. So we can't really put them on a light microscope easily, but we can put them on the stereoscope and get a good look at the actual cells and cellular structure of these things, right? So you can see that here, I hope. Right? Pretty clearly. Really pretty looking plant when you look at it up close. From a distance, it just looks like a lump of green stuff, but the really soft appearance that moss has, that we sort of think of when we think of mosses as being like a soft place or a soft thing that you can step on. But, in practice, it's actually a bunch of little cells. Okay. What kind of objective is it? So this, as I mentioned, is a Leica stereoscope that we use. It's the somebody hit that button up there that actually tells you the name of it. It's the S9i, and I think it's achromatic is the name of the lens, the type of the lens, but. You can look up the details on if you just Google the S9i for Leica.
What were the first types of plants to come from the primordial soup?
Yeah, mosses and like liverworts. None of them came from the primordial soup. The first organisms that came from primordial soup were cyanobacteria, very likely. But the really early plants, like the very first plants that arose, very likely were like the liverworts. And we could get some liverworts. I find them around here growing in sort of like wet walls and things. And I think one time I tried to put some on the SEM, but maybe I didn't. Well, I can get some for a later stream. We can talk a little bit about the liverworts, but they're pretty cool. Very simple type of plants.
We find on exoplanets.
On other planets?
Yeah, exoplanets.
Probably not plants. If we find life on the exoplanets or the planets that are way out on the edge of life, they're very likely it would be very different than life on Earth. But it's so far away from the sun. Probably most of those organisms would have to be chemotrophic rather than autotrophs. So they probably would use some sort of other type of energy to make nutrients. And we have chemotrophs on planet Earth in the hydrothermal vents under the ocean, where they're super deep and dark, where they basically use nutrients from hydrothermal vents, and they might, like that sort of organism might do okay in those kinds of environments. If there's hydrothermal vent-like structures, it wouldn't be like life on Earth, though. It would probably be something very different if it exists.
Did you find any sporing moss?
Did I find any what?
Sporing moss.
Oh, sporing?
Yeah.
I don't know. I don't know what sporing moss is. Who was that question from? Jurgling? Sorry, I'm not a moss expert either, guys. Probably people who've been to my stream a lot know that I study diatoms, and thus the name. But I also just kind of am generally a naturalist and study lots of things. And if it fits under my microscope, I usually like to look at it. So I'm not an expert in mosses, or lichen, or moths, or pick something. I'm probably not an expert in it if it's not microscopes or diatoms or SEMs or maybe photography. But I think it's fun to still show these things to people. So I don't know if those are sporing or not. I presumably those are some kind of pollen grain on the actual moss that we're looking at right there.
Why are they green?
They're green because they have chloroplasts, right? They're autotrophic. So the green is a pigment that allows them to capture the right bands of light from sunlight and use it for generating energy. Yeah, those look like some sort of pollen-like spores. Blink balance. What are you up to? How you been, Blake? I haven't seen you for a while. How's your streams going? Can somebody give Blake a shout out if they haven't already? Oh yeah, there you go. Yes, I lead a diatom army occasionally to raid other channels. But if you're going to ask me tough questions, you're going to get a lot of like, I don't know. I like looking at cool things that are very small. So, and showcasing them for other people. I haven't seen any antenna-like structures, Jurgling, but if you see something and you know what it is, we can rely on other people's expertise in the channel, and you feel free to explain to us what we're seeing. That's fine. These look like, I don't know, they look like some sort of little pollen. They could potentially be pollen off of the tree, I suppose, that the moss was collected from, though. I can't see the pollen close enough to actually tell what it is. I suppose we could slap it onto scanning electron microscope. I have some other things to look at while we're looking for pollen. One of them is, for some unknown reason, this... Oops, let me zoom out. As long as we're looking at little things with pollen on it, this is actually a dandelion.
There are
And
2 more questions. Boss.
2 more questions. Throw them at me.
Okay. What benefit does the fluorescing provide? What is
The fluorescing? I don't know. I'm not sure what you mean. The, there's some value to it for them to convert energy, I'm sure. What's
that extra light? Is that extra light also drawn to energy?
Oh, maybe. I doubt it though. I don't know if do mosses actually fluoresce. Is somebody saying that they give off a light? Maybe there's a value for it. Um,
And another one asking why it's looking yellow something. Yeah, why it looks like yellow.
Why does it look yellow? Well, I switched us out from the moss to a dandelion. This is a flower that grows in North America. People think of it as a weed, but it's really a flower. And normally in the spring, but for some crazy reason, I was out walking around and I was like, what the heck? There's a dandelion. So I just grabbed it and brought it in. I thought it would be kind of fun to look at. So here we are looking at dandelion. And this one I could probably do a little bit better telling you parts on it. So you can see these little curlicue things right here all over it. Sorry, let me adjust the light a little bit. Try to center the light so you can see these little curly things are actually the sexy bit of the plant so that it can reproduce. And it does hold the pollen and is a place where the pollen is also deposited from neighboring organisms. So this is, again, zoomed as far out as we can go. And I can get us a lot closer. We can see the little bits of pollen that are actually here on the dandelions, sexy bits. And this is the stuff that gets collected when a bee lands on it and tries to get a little bit of sugar out of the plant to convert into honey. So it will land on it, and then this sort of yellow junk here is the pollen. So if you're allergic to dandelion pollen, this is the stuff that gets into your system that your body doesn't like. And if you're a bee, it gets trapped on your legs and your butt, and your hairy bodies, and gets carried from one plant to another, where ideally it then ultimately gets germinated and allows the plant to reproduce, right?
Is pollen a lipid structure?
Pollen is made out of something called sporopollenin, and it's organic material. I don't know if it's lipids. You keep asking me biology questions and you're going to get a lot of question marks back. I don't know the answer to biology questions like what's the stuff generally composed of. But the pollen is basically how the plants reproduce, of course. So, and you can see those sort of really cool little curlicue shapes. They usually come up from the middle and go out in both directions, and then the pollen that gets transferred onto the plant from bees basically has to find its way down those long tubes that the two little curly things are sort of stemming from, and become available for the plant to germinate. Know all the answers, please? I will if you will, Blake. We can't put every single type of thing under a microscope that fits there and have me know everything about it, unfortunately. There's a limit to the things I know. I was trained as a geologist, so if you have geology questions, I can answer those, pretty much all of them. If you start answering me biology questions, and it doesn't deal with diatoms, and even sometimes when it does, you'll get, I don't know. I'm showing you these because it's fun, not because I think you're going to learn a lot about dandelions. But one thing I will say is that people think of dandelions as being just like a weed. This is a nuisance for some people. because it grows very well in lawns. But I think they're pretty, and I actually think that they're really necessary for bees. They get a lot of pollen from these and make a lot of honey. So they're actually a valuable component to a typical yard ecosystem. So if you're going to have grass, you might as well have these as well, right?
So, what is obsidian in Minecraft made out of?
What's obsidian in Minecraft made out of? I think you take lava and you mix water together and it makes obsidian, doesn't it? That's not actually how it works in real life, but it's kind of close. Obsidian has got a glassy texture because it cools very rapidly, and when it cools very rapidly, it basically makes disorganized glass, and that's why it's so sharp, and also why it has conchoidal fracturing. So how'd I do? All right, so the composition for it is it's got a lot of silica in it, so silica and oxygen. Cool, right? So pretty neat view. Again, this sort of middle scale that we're looking at, we can get a lot of sort of cool things that I'm not used to looking at in color. So we can put these things in the scanning electron microscope, but of course something like a dandelion would not do super well because it's got a lot of moisture to it, so it would be dried out basically. And we could look at the actual petals of the flower as well. I was trying to get some into focus. It's kind of a challenge because it's shaped like a cone, and I just have to smash stuff in order to get it to get into the camera to give us the view that I want. And I'm going to zoom out a bit. There we go. So you can see the actual petals of the plant, of the flower right there, because we're kind of way into the middle of the center of the flower where all the pollen and the sexy bits are, but there's actual little petals and sepal. What was it?
Diatom Lord, you are.
Diatom Lord is looking at dandelions, and it's minus 20 C out right now. I don't know what to tell you. There's some dandelions. Yeah. Oh, I see my wife has showed up to answer questions about obsidian for you. So Campley Discus is actually my wife who's tuned in from home, and she's also got geology training, is teaching a geology class right now. So if you have some important questions about geology, now's the time to ask them. There's 2 expert geologists in the channel. Maybe Volcano Doc will also show up into my stream. You never know. And then we'll have all kinds of questions we could answer about obsidian or volcanoes if you'd like. Maybe give a shout out to Volcano Doc, Pacific Plankton, because if you really have a lot of questions about volcanoes, you should talk to her. And she has a nice stream, just geology here. She would be a good person to sort of tune into if you have a ton of mineralogic or questions about obsidian or volcanoes. I'm just going to tell you, though, I don't think she knows anything about Minecraft. So if you have Minecraft questions, what you should do is ask those, and then my wife can ask my daughter, and she could answer pretty much all your Minecraft questions. There's also, on the bottom end of the flower, what are called sepals, and here they are, for dandelions, they look something like this. So you can also see these like long hair structures that they have that are in between the sepal and the petals. And I think I got a little bit of the stem on this one. Maybe if I flipped it over. Dandelions have really kind of interesting stems. So they've kind of got like a little hair all over them. It's the center thing that's kind of orange is the stem, and the plant's not moving. That's my finger trying to hold it because it's not going to stay in focus otherwise. But characteristically, if you break open a dandelion and kind of squeeze it a little bit, there's sort of like a milky substance that's my finger, a milky substance that kind of comes out of the stem. And the stem actually is a hollow tube, like a straw, so kind of cool looking. Multidisciplinary family. Any questions you have about Roblox or Minecraft, my daughter will take. She'll address all of those. So if you have pressing questions about any of those things, she can give you a really good answer about all of it. Let's see. So we've been looking at some plants and some insects. Why don't we look at some components of vertebrate organisms? So you can start asking me questions about something else I don't know anything about or know limited things about. Presumably you can guess what this is based on what little bit of it you can see. Right there is a feather, of course. This is the plumage. We're just looking at an individual feather. And I have a funny story about this feather. So here's what it looks like, right? I found it out in the same place where I was looking for the lichen, except for there was just a pile of feathers on the ground. Like, there was the tree, and then there was like a pile of feathers, and the bird was missing. And I'm just going to guess that this thing got eaten by a hawk, because I don't know what else would cause a bird just to have feathers with no body laying around. I suppose it's also possible that there's a wild cat on campus that somehow managed to get one. But I know that there's hawks on campus and that they are pretty active at grabbing up the birds. So I suspect it's a robin. There's a lot of robins on campus. And
What campus is this?
it's Indiana State University, which is located in Terre Haute, Indiana. And this is my lab. This is actually a paleolimnology lab. We study lakes and sometimes wetlands and sometimes rivers, and sometimes just really old geological deposits that have lakes or river-like substances in them. And there's a little bit of red in here. I'm trying to figure out what that could be in the middle of this thing. Let's see if I can zoom in on it. Of course, we can do that. And you can get a good sense of the plumage on this feather. And I don't know what that thing is. I thought maybe if I zoomed in, it would answer questions, but it just created more of them. So we're going to do this. You never saw that. This is not the red thing you are looking for. And I don't know what it is. Earlier, when I was sort of scooting around on this, looking through the microscope at it, I found some pollen, like a pollen grain or two, trapped in the feather of this bird. Let's see, get out here towards the end, it becomes a little bit more, it looks a little bit more like a bird feather. Oops. So for now, the little piece of plastic that we use to let us know where the center of the stage is kind of blocks it, pokes at it. Let's zoom in on that.
Someone asking, is it plastic?
It's not plastic. Oh, the red? That red thing might have been plastic. That's not an impossible. It's a microplastic. So when you get zoomed in again, we can't really get the whole thing in focus at the same time because the depth of field is very small when you're zoomed in like this. So, but you can actually see the little individual plumage on the feather, which is kind of nice. And then early on in the stream, I was playing around with our microscope's ability. So our microscope has a really cool superpower, which is that I can actually make the rest of this image in focus if I want, by just focusing and starting the Z stack. So I can do this. I can start, start the Z stack by pushing this button. Oh, unless I turned it off. No, I didn't. Okay. And I've got to give it a file a name, or it won't leave me alone. I don't know where the image went, though. just disappeared. Guess I shouldn't have played with that button. No, it's messed up. I did something and crashed it, maybe. I didn't like it. We'll just ignore that for now. See if I can make it work now. No, this thing. Ah. So, I think what I can do is, as I focus, different parts of it will come in to focus. Oops, that's the way it's supposed to work. Let's see. Once it starts.
Any diatoms on the feather?
Are there diatoms? Who's asking for diatoms?
I think Dr. Westover.
Oh, her. Well, we were looking at other things first, but if you want to look at diatoms, we can do that. Hang on. So, not on this feather, right? So you can, I haven't seen any diatoms on the feather. I did find some pollen somewhere in here, but mostly it's just the texture, right? So you can see the vein of it, the texture of it, pretty easily. Okay, let's swap it out for, let's see. We could look at one of these sweet gums. Don't ask me questions about sweet gums, Blake. I don't know the answer to sweet gum questions. It's a seed of a tree. And I need to really zoom out a lot because it's a lot bigger than everything else we've been looking at. And also, too much of my screen is out of focus. So what we're looking at now is a little sweet gum. This is, you know, you see them hanging off a tree, sweet gum trees, and then they just kind of sit around on the ground. They can look like this too, right? This is the red one. This is the old one. This is a new one from this year, probably. And we're just giving you some examples of things that we could see sort of mid-size. There we go. It's a nice view of that surface. We keep trying to put these things in the SEM to look at it because I always think they're going to look really cool. But the thing is that it's hard to get something like this in the SEM because it's a giant ball. And it's hard to mount it correctly. And even if we could get it mounted correctly, it probably, you know, it's just going to look like maybe a little more boring in the SEM because all the color is gone. But it was kind of a pretty cool view of it. Some of the bright colors that you can see on it. And it's, of course, covered with dirt. So you can see little specks of silt and sand on it. And a lot of that sort of three-dimensional structure that's in it, right? So we can get a pretty good idea of the three-dimensional component, as I can only get a little bit of it in focus at the same time. Now, I feel like I could risk it again. I could try to turn on the Z-stack and make it work this time. Who knows if it will? Give it some crazy value. And I'll try to focus through this thing. There we go. So this is what the Z-stacking does. The stuff that we started in focus was up here, and the stuff down here was out of focus. And now all of it's in focus. I made it work. See? Just a little bit of practice, and then bingo. And then we get an image out of it where the whole thing's in focus, right? But then you lose the sense of dimension. You lose the sense that it's a three-dimensional object, and and that the spines are sticking way up into your face. But you then have the whole thing in focus all at once, which is kind of cool. So again, that's a really neat feature that comes built into the software associated with this Leica stereoscope. It kind of creates these really interesting, sometimes interesting views of things, where rather than seeing just a little bit of it, you could see the whole thing. But if you were going to publish that, of course, there'd be a problem, because it doesn't look like that to anybody else. It only looks like that when you have everything in focus, which just can't happen very normally. So normally you just have this sort of really strong depth of field. All right, made it work. This is, what else do I got? Got a little leaf that we could look at. So dead leaves laying around are pretty easy to find. Have any questions over there? No questions? Good. I love it when there's no questions about things I don't know anything about.
But someone asked that do diatoms pass through the digestion still alive?
Can diatoms do what?
Do diatoms pass through the digestion that's still alive?
Yes, diatoms can sometimes pass through the digestion process of things like fish and come out the other side living. That can happen, for sure. Most of them probably don't, but some of them definitely will pass through, and they'll probably come out a little bit happier because there's a bunch of nutrients associated with the poop. There you go. We thought we were going to look at something boring like a leaf, but when you get in really close and look at it, you can see the cellular structure pretty well. And that's kind of a nice view. We can also zoom out and see the sort of reticulated structure of the veins on the surface of it. In the scanning electron microscope, if I were to put the same leaf under there, we would see mold spores and bacteria and all kinds of other things that are growing on the undersides of these. The leaf is being decomposed, so our decomposers would basically be taking over. You can see it a little bit in here. Some of the larger strands of stuff that you see that are kind of like a dark orange color right there. Those are probably bits of decomposing material that are on those leaves and causing it to break down.
Is it possible to see fluids flow?
Is it possible to see what?
Fluids flow. Fluid.
Oh, fluids. Well, everything's dead that we're looking at, so, but yeah, we could see fluids moving in the, if it were living. You know, it's kind of hard to fit the whole live plant or a whole live something with fluids in it, but oh, here's an example of something we found growing underneath the leaf, right? Some things that have basically colonized the surface. That surface is probably just covered with mold and bacteria that are working to mineralize it, right? So I think here's some mold down here in this zone. Let's see if I can get the focus right. You see those sort of dark patches there on the underside of this leaf. Probably a bit of mold growing. Let's see, pretty good view of it. All clustered together in one little place.
Do some vasculature have pumping mechanism in planets?
What's the question?
Do some vasculature have pumping mechanisms in planets?
In plant? Oh, in plants.
Planets or plants?
In plants. I don't know. You're trying to trick me with questions about biology again. Probably there are some. Maybe Sarah's in the channel. She's a biologist. She might be able to answer those questions. They probably... Oh, I think she did try to answer that question. It's mostly capillary action, but there probably are some that can kind of force the situation a little bit more. For most plants, though, if you think about it, the leaf is out here, and water is evaporating off of the leaf. And as the water evaporates out of here, the little straw-shaped structures that are leading to it basically have a vacuum that's created by that. It'll pull up material, mostly through surface tension, and that basically wicks the water out of the plant. So let's see. I got a pile of things I've been looking through.
And Carling is saying you should do one of the Sylvia's baby teeth next time.
Do one of the what?
Sylvia's baby teeth next time.
Sylvia's baby teeth? I guess I could. And now she comes up with ideas for me. She's like watching the stream again. She's like, you should do something cool like baby teeth. This is on the other side of the leaf where these little buds. So this is little buds. I saw the Pacific plank. Do I have any fuzzy leaves? I don't have fuzzy leaves, but I have some fuzzy buds. Here's some fuzzy bud. A cute little bud at the end of this tree's branch. This one, unfortunately, was on the ground, and so the whole branch part, the leaves and the buds had been basically broken off. My guess is it's a squirrel that was responsible for it. But we can get zoomed in on the fuzzy bits of the bud. Here you go. If you like fuzzy things. That's nice. So get a nice little view of those. The bud, of course, is where the leaf or the branch continues to grow and the leaves come out. What else do I have? Oh, I do have some. I made a little slide. Last night I was doing a stream where we were looking at water bears and some other aquatic organisms, and ones that are found in Lichen. And I had scraped some of the Lichen clean.
Okay. So, someone is asking, hey Professor, how are you doing?
Who is it?
Headshot specialist.
Hey, Headshot, how you doing? I'm doing well. Someone could maybe give Headshot a shout out. There's the same view of the things that we were looking at in the light microscope last night, but now I can see them in a stereoscope, a stereo microscope. And for all the times where I could never see what was on the other side of a lichen, now we're on the other side of a lichen, and we can actually zoom around and look at the slides where we can actually see the opposite side. So this is kind of equivalent to looking at a microscope using dark field, where we can basically see reflected light off of a surface. And dark field's usually got a lot darker component to it, but this is more or less comparable to that, where we can see all the pretty colors of what we're looking at, instead of what we normally get, which is outlines of lichen. So here we can see, just from the sample, some of that material. And if I was really careful about looking around in here for long enough, of course the problem is I have to move everything with my fingers. We probably could find...
So someone is asking, what is the red in the background? I think that plastic, that microplastic.
Oh, that little red thing is just a little tab that we use to figure out, to point to where's the middle of this the stage. So the guy who set up the stereoscope stuck it on there while he was trying to get it lined up, but it's just like a sticker, like the kind that you'd use to sign here, right? One of those. So not particularly valuable. I could always take it off, but I always just leave it on there because sometimes it's hard to find the middle of the stage.
And another one is, can you do dark and light fields together?
I can't. This is the only light type that we can do. The only thing that would make it more like dark field is if I made it more oblique. So if I actually just totally tilted it now. It's more like a typical dark field where the angle of the light is more oblique. But generally speaking, you're either looking at something in dark field or you're looking through it in bright field. So I suppose you could turn them both on if you had something like this structure where you had a top and a bottom light source at the same time, but I don't know that it would be beneficial. Not something that I've tried because I haven't had a microscope with dark field. I usually do things in bright field or differential interference contrast or phase contrast.
Okay. Do you have light from under the plate?
No. A stereo microscope is meant to be used with reflected light, not transmitted light. Hey, I found a nematode. A bunch of them, actually. Probably as we heat up the water, they're starting to get excited. We can zoom in.
Can we look at a CD?
A CD?
Something with spectral scattering.
Can we look at what?
At a CD.
Oh, a CD.
Yeah.
What? Like
Yeah, the disc.
a compact disc? What are you, from the 80s? Yes, we could look at something like that. If I could find one, I don't know. I'd have to find one in an antique store. Yeah, we could definitely do that. Sorry. I don't know who asked the question.
I don't know.
Zoom in.
That type of microscope exists where we can do the dark and light field and then uses computer synthesis to combine the two images.
Okay. I don't have any use for it, so just because it exists doesn't mean it's something I need.
Like, he was asking, does this exist?
Oh, does it exist?
Yeah.
I don't know. Again, I don't need it, so you could take two pictures, though. You know, like a picture. If you were taking an image, you could take a picture of using light field and then a picture using dark field, and you could combine them pretty easily in Photoshop. Those just make them different layers, and then you could.
Moving your plate.
Yeah, yeah.
What
These are these are nematode worms. So there's a nematode worm here, and there's one up here, and there's another one over here in the background. I was actually looking to see if I could find some water bears because rotifers and water bears should be in this sample. And we found some last night on my light microscope stream. So, but they're always, well, not always. I usually have to hunt for them. Last night we had a really funny situation where a bit of the water was pouring off the side of the slide, and a water bear just happened to roll right through the middle of my field of view while I was looking for water bears. So I didn't have to move it, and I got one just to come visit me. So it's nice when that happens, but pretty wild.
Okay, what's the green stuff?
All the plant material that you're seeing in this field of view comes from lichen. So for this sample, what I did was I just took a stick and I scraped it with a knife. And it's the lichen that was growing on the stick that we're looking through. So all of this sort of green and brown stuff that you're seeing is organic material that's come from a lichen. And some of the lichen have this sort of white color as well. And there was also a little bit of mold in the sample growing on the tree, I think. So there might be some mold in here in some of this material, but so that's lichen, that white, bright white that you see right there, and the green is also lichen, and I found a rotifer.
Do you think we can observe their growing by mitosis?
We can see organisms dividing through mitosis for sure. If they were dividing on my screen, if I had a diatom or an amoeba or something like that that used binary fission.
What are your prophecy?
This is a rotifer. That's a bedelloid rotifer that you're looking at right there. So in our view that we have, see if I can get it to light up. We're zoomed in as far as we can on this stereoscope, so this is as close as I can get to it. But that's actually pretty close to what we could see, like I said, in a light microscope at maybe 20x or somewhere between 20 and 40x. That's pretty much how it would look. So rotifers, the bedelloid rotifers are sort of extremophile type organisms that lived in the lichen, and then when the stick is dry, they go into like a hibernation state the same way that a water bear does. And then when I re-wet the sample last night, I just kept it wet by putting a lid over it, and then so the rotifer's been happy living in this sort of soup that I made with the lichen fragments and a little bit of water. So and again, if I were to let the sample dry out and then put it back outside, every time it rained, basically they'd come to life, and every time the environment would dry out, they would basically go into sort of hibernation. So, and rotifers attach with a sort of a claw-like foot that comes out of the bottom part of their body, and then at the top part of their head area, they have like a feeding apparatus called a corona that they use to funnel water towards their mouth with the nutrients and organic matter that they then consume. So we often find them in our samples with the water bearers because they have similar habit. And so while it's not a water bear, it's a good sign that we might find water bearers. So
Someone is asking, are you looking at a stick?
I'm looking at the scrapings from a stick. The stick is gone, and it's been mounted onto a microscope slide because I wanted something that was flat and clear that would allow me to see into the sample. So it's the scraping from a stick. We could actually look at the stick, but then we'd have to wet it probably, and then I'd have to have the stick under here, and it might be a little bit more challenging because of things could be hiding inside bark and other stuff. So in the light microscope, they don't have that option. They're just going to sit and.
How did the Gomfa monsters go?
How did the Gomfa monsters go? So far, so good. We're still working on characterizing them, but on Monday I'll be getting a sample from the guy who shipped it to us. That sampled it.
Good news,
asked
everyone.
randomly if somebody would send me some material from a site, and they happened to be nearby, and they just went and got it for us, and they're shipping it to us, and it will arrive on Monday, so we'll see how that goes.
Can those nematodes we see here be spotted by the naked eye?
That's a good question. I'm going to say no. Not by my naked eye, at least. It's a little... What?
It's not possible for me.
Not for you either? We're going to go with no. Maybe if you had a magnifying glass or something like that, you could potentially see them. What do they do with all those slide glass?
Somewhere here. Oh, here.
Oh, thank you. Perfect. Sorry, I wanted to make another sample out of this material that I collected. So yeah, it's like a little stew. That's from the stick. I just sort of added some raw pure water to it, and so you're looking at just a few drops from this, and I just take a little pipette and pipette some of it up into a transfer pipette, and then just dropped it onto a cover slip. So that other one we had was starting to dry out, and also I looked through most of it and I didn't find any water bears, so I thought maybe if we really wanted to see a water bear, if that's why you came, you're going to be disappointed if I don't find one. Then I thought maybe I'll just look around a little bit in some of this material a bit more, and maybe we'll see something. So it hasn't been sitting under the microscope, and it hasn't been drying out quite as much as a result. And it's possible that there's a water bear in here that's been wandering around since last night eating material. Oh, this is actually kind of nice because right here, let's see, where is that on this? Oh yeah. Right.
What are we looking at?
It's just another sample from the same stuff. It's a lichen, a slurry of lichen and water, and from a stick. And actually, I wanted to zoom in and show you guys the actual lichen. They're called squamules, which are the surface of some lichen. They make sort of these polygons, and they're right there at the bottom of the screen. This sort of like little hexagonal shaped components. I think you can see my cursor right here. These things are called squamules, and you can actually see that the squamules is a whole cluster of them right here that are on these lichen. So some of it, while I was scraping, didn't get destroyed or destroyed too badly, so you could actually see those. Just trying to move my seat down a little. So we could look through here and potentially, because one of the things that's nice about being at this magnification is it's about the right scale for picking out a water bear pretty quickly. So I can kind of scan around in the material. In the light microscope, oftentimes I have to look a lot longer because here it's easy to detect things that are moving, and like if we saw a water bear crawling around, we might be able to pick it out a little bit more readily than in a light microscope where we're zoomed in a little too close. This is good. There's another little rotifer hanging out right there. I don't know if you can see that yet, but I'll try to make it apparent to you. There he is, hanging out on that floating piece of debris. Get a pretty good look at him crawling around looking for food or repositioning itself. That's again just another rotifer, same type we saw before, maybe not the same species, but it's a bedelloid rotifer. It's in that family. That's a very small one. The other one we saw was actually probably about twice as big as that one.
Are there computer programs that help you spot moving things under the microscope?
I don't know. I usually just rely on my eagle sharp eyes to pick things out because I'm used to looking at small things in the microscope. And I also have the advantage because I have to move this around with my hands. I know when I'm moving it and when I'm not moving it. So it might not be obvious to you how I got from like something that was shaking to, oh, there's clearly some moving there, because I know when I move it, it's supposed to move, and when I'm not moving it, it's supposed to either wobble a little or stop moving. So it's a little easier for me to pick out
Good news, everyone!
the movement of particles. It'll be kind of interesting if we find a water bear. They'll be roughly the size of the things that we've been looking at that are moving around in here. And they might have gone back into their like sleepy stage. It's hard to say. The sample's been wet since last night, and maybe they're not used to being doused in water for that long. There's a nematode right there that's not moving. Also, it's very three-dimensional, right? So we have piles of stuff all over the place. It's possible that it's crawling in and out of some of those piles and I wouldn't notice it. So I see a little ciliate moving around, which is kind of crazy at the scale. Any more questions I could answer?
No, not yet. No
People,
question.
people are just fixated on me creeping around in here. Okay. So I didn't find anything. We can come back to it if there's a little bit of time. It's 2:30, and I wanted to actually showcase some other things. So I'm going to have to clear off a little bit of space here and move over our Micro Manipulator. I don't want to crush any of my samples from today. So the piece of a moth doesn't want to leave. Just going to set those right there. So I've showcased this Micro Manipulator before. We're in the SEM lab. It is effectively just a series of components that can be moved in different directions. So let's see if I can get this so you can see it. It has at the base a set of rare earth magnets and a switch that basically turn on and off really strong magnets. And then I can set the height with this knob. I can then change the movement in and out of in the sort of X direction with this knob, and then with this knob up and down, and with this knob in and out. So right, this is Z, this is Y, this is X, and so I have X, Y, and Z. I can move in all the directions I need to, and then on the back side, there's a little joystick-like thing that actually lets me move like I could actually move and nudge things in small dimensions a little bit. So if we found something and we wanted to move it or pick it up, we could do that with this. We just basically get it to the height that we want. We set all of these things around 0 so that we have the ability to, helps if you turn on the magnet, set these things around 0 so that we have the maximum amount of movement capacity, right? So we can move forward or backwards now very easily. And then I can kind of align it where I need it to go. And then from there, we could use it to basically move very carefully in and out. And on this one, I've mounted to it a little sewing needle, which isn't the typical specifications that people would use with one of these things, but I don't have any of the glass that you need, so I just winged it and I stuck a needle on there, and it'll actually be nice because it'll give you a sense of how big what we're looking at is relative to a needle. And in most cases, everything that we're looking at is very small, and so the needle is going to look massive in our field of view. So this is a sample. Oh, I should look and figure out which one it is. These are from San Francisco Bay. So these are some materials that Pacific Plankton sent to me, and we put them on SEM stubs, and we're actually looking at the surface of the SEM stub. Now these have been coated with gold, and so there's diatoms and other debris that are on here that you're seeing in their normal sizes. So again, it just kind of looks like dust at this level. And here's another one. There's a bunch of stubs in here. They all basically have the same kind of stuff on them. San Francisco Bay material, and hopefully some little tiny diatoms. And what I'm going to do is get some light on the subject, try to get zoomed in so we could actually see some of these diatoms. Now, marine diatoms tend to be big relative to freshwater diatoms, and particularly the plankton are pretty large. So, these are unusually large diatoms that allow us to do this sort of thing with a needle. I probably would need to get something more small otherwise. What you can see, hopefully, are these little things that look a bit like diamonds all over the surface, right? See these highly reflective
Good news, everyone.
diamond-like structure everywhere. There's a really bright purple-looking one. So that's not a real purple. That's just the light being focused through it like a prism. And for some of these, I believe you can actually see that it's clearly a diatom. Hang on. So hopefully you can get that, the one that's in center right there. Let's see, let me switch to this right here. That's a diatom. These are all diatoms, these little round things. This is a really interesting view of diatoms because normally we see them either in the scanning electron microscope or in a transmitted microscope where you're seeing through them. But here we're actually seeing the surface of them, and can in some cases very clearly see the surface of them, right? So there's this big one right there that's right in the center, and a whole bunch of little ones all around it. They're all a little round, bunch of little round guys everywhere, right? You can see the light coming off of that one. It looks like a little drop of water or a tiny diamond, but in fact it's a diatom. So kind of cool that we can see diatoms at this scale. Even some of the smaller ones are visible here to us. All the round ones are really common in the Pacific material, in the San Francisco Bay material that Pacific Plankton has sent me. And so we can see a whole bunch of little round guys. I think we could probably, I might even be able to identify some of them. So you're asking about reflective things that look like a compact disc. You see that one right here. I believe that's an actinocyclus. And we can actually, I can kind of tell what it is from here because of their quadrant, sort of reflective quadrants. So I think that's really cool. I'm pretty excited to be able to look at diatoms in this weird reflected light sort of technique. So there's a dark one right here. I think that's probably a Cosinodiscus based on the surface texture. And not all diatoms are round. I hope to disabuse you of that idea. There's some other things that we can see in here, like, let's see, right here, I think that's a diatom. A little stack of Ellerbeckia, maybe. And then, oh, look at this. Isthmia. Look at that Isthmia. I hope Pacific Plankton is here. Is she still here?
Yeah.
Isn't that amazing?
Yeah.
You can just look at that Isthmia. It just looks like a thimble or something sitting there, right? Just huge and gorgeous, and we can totally see it in a stereo microscope. I'm so excited by that.
What is this in the genus like?
Isthmia. It's Isthmia. Isthmia. It's the one that's actually the little. If you have access to the emotes, it's this one. So we're seeing one valve of it, a giant Isthmia valve, and I'm gonna pick it up. I'm gonna get crazy and try to pick it up. So we need this thing. Perfect. And then I need one of these things. They can't see what I'm doing because I'm not on the camera anymore. I'm just prepping it. So normally we put these sort of discs on the SEM called stubs. This little thing, and it's covered with carbon tape. And I've lost my Isthmia, but I need to move it anyway, so that's not a big deal. Let's see if we can find it again. It's big enough, easy to spot, right? So there it is. Super cool. Now if I only had a tiny little necklace, I could put that on, you know? tiny necklace and dangle it.
Yeah.
All right. Let's see how we can do with the micro manipulator. So, I need to unlock it, rotate it around, and I'm going to get the arm kind of close. And then we will see how well I can do with it. All right, so you see the needle? Way up here, up high. And I need to first lower it to get it kind of close to the stub. So for clarity, that is a sewing needle, the tip of which is now visible in our field of view. And our isthmia is a little bit this direction. I'm just turning the knob to move it a little bit closer towards it. And then this direction. Now that I'm a little closer, I can zoom in. That is what isthmia looks like next to the head of a needle. So the needle, you can see, has a little bit of junk on it because we've been doing this before. And I'm going to lower it just a little bit. Now it's actually touching the material. You can see there's a bit of an electrostatic charge. I'm picking up some diatoms by accident. I'm just trying to get it a little bit closer. Nudge it. You can see I'm making kind of a mess out of the bottom part, but I need to get a little bit more in this direction. Boom. I just picked it up. Right?
Yeah.
You can see that?
Yes.
So I'm going to hopefully keep it on there. I think it's still stuck on there. And I'm just going to slide it over to the little piece of carbon tape and lower it down. So I'll zoom back out a little bit. I'll see if I can get it in focus for us. It's still there.
Yeah.
And it needs to go a little lower. You can see the actual tape getting sort of torn, but and it's sticking a little bit to my needle. Boink. And that, ladies and gentlemen, is a micro manipulator. So we went in, we found what we wanted, we zoomed in on it, and now we have it right where we want it, which is on its own little stub, separated from everything else. There it is. Perfect. So if we wanted to
Good
flip it
news,
over,
everyone!
or set it up on its side, or do any number of things with it, basically we could go in, pick up a diatom, and transfer it onto our tape. Just using a little bit of the electrostatic charge of the needle, and that was it. So I'm going to play around with the Micro Manipulator later. I don't know that I want to sit here and try to pick up a ton of diatoms with it. You saw the sort of thing in practice. You got to see me do it, so you know that everything was being done live. But I will tell you that if you wanted to pick up smaller things, you'd need to have basically some really fine fiber optic sort of 100 microns or less sort of thickness glass. So if we want to get some of those really little ones, we could potentially do that. And of course, the more you use the Micro Manipulator, the better, the more deft you become at actually like manipulating things with it. But that's the general idea. You can pick stuff up, move it around.
What is this? I think he's asking the name of this diatom again.
Oh, the name of the diatom is Isthmia, and the Micro Manipulator is a manually operated one. They do have, the whole system is just this. They do have ones that are used with little step motors that can be used and controlled more like an actual joystick on, like with a game sort of thing where you can just push X, Y, and Z and it will spin a step motor and move things around for you. I should point out that in the old days, prior to, like back in the Victorian era, people would arrange diatom slides. They would pick up diatoms like I just did, using a similar device, but not nearly as well constructed. But, go back to the Victorian era, and basically they would have these sort of micro-milled gears. And at the end of it, rather than using spun glass, they would use a pig eyelash because they're very fine. And it turns out I don't have any pig eyelashes, so I couldn't use that either. But you could use the pig eyelash because it has a very fine tip on it, and they would just pick up diatoms and rearrange them and spell things out or draw pictures with them. They'd have a collection, like I do, of different types of diatoms. And on this SEM stub we have, and they would use optics probably kind of comparable to these, or they would use an inverted light microscope and their little pig eyelash, and they would pick them up and place them on slides and basically arrange them into these really cool artwork. So people...
You're requesting to do it again one more time.
What? Oh my gosh, I got to do it again? Just once wasn't enough for you. Okay, I'll do one more. I can try to pick up a little round one and put it next to it so it has a friend.
It was like a fine operation.
What's that?
Fine operation, like OT. You're doing operating something.
Oh, yeah, it is like it is a little bit like a doctor operating on something.
Like my appendix, they cut off.
In fact, modern doctors use microscopes quite a bit, I think. My wife had surgery recently. I think all of the surgery was done on a microscope or with a microscope. All right.
So, do you have the same ability to manipulate in the SEM?
No. I'm actually putting them on an SEM stub so we could put it into the SEM if we wanted to when we were done with it, but I don't have a micro manipulator inside the SEM. I will say, though, that they do make micro manipulators that fit on and into SEM chambers that allow you to do the same thing inside the SEM. I don't have one of those. I don't, this is, it's not a toy. It's used for me, although I'm using it as a toy right now. For me, its use is actually that we might want to pick up an individual diatom and move it around so that we could position it in a way to see something in the SEM. So for that, it's actually really valuable. Right now I'm just kind of showcasing it as a bit of a toy, playing with its ability to pick things up and move it around, but it's actually good practice for me because we haven't had it up and running before this point, and so my skill at moving things around is not, quite as good as it could be. So my undergrad student, Rihanna, is working on a project where we need to pick up some diatoms and move them around and look at them in the SEM. Rihanna was here a little earlier. I don't know if she's still here, But, so she's been learning how to do this. I don't know what that big glowing thing is that's sticking up at me is.
Can you pick the red one?
Can I do what?
The red one.
The red one?
Yeah.
Somebody's asked me to pick up like a certain
No,
one?
I'm asking.
You want me to pick up a red one?
Yeah.
They're all covered with gold. How do I know which one's a red one?
I also missed.
Oh, is this sort of ball that's here in the middle? Is that what you're talking
like, red and black alternating colors.
about? Yeah. I don't know what that is. Let's go see if I can knock it over. Let's see. I need to go
Here you have one, the black one, the big one.
down a bit. I need to zoom out so I can figure out where my needle went. It's like, do your trick again. Do the trick. Okay, fine. I will do the trick again.
Someone is asking now, what do you frame them? Do you frame them on the wall?
I suppose you
You
could.
can remove this 2020 calendar and
I suppose we could. I don't think they would be very good on the wall as a wall art. I need that to move up, and it was moving down. I was pushing in the wrong direction. I'm not sure what this thing is that you're talking about. This is the red thing that you were interested in, right?
is it?
I think so.
I think red one is black one.
None of them are red. They're all gold. They've all been coated with gold. So I thought you meant this thing. Is this the thing you were talking about?
No.
I don't know what that is.
Okay, just pick up this one, the big one.
That big ball?
Yeah.
I can pick it up.
No, not it.
Oh, you mean
That. No, again it.
you want that one?
Yeah.
Okay.
You didn't pick up that.
I broke it.
No.
Oh no, I picked it up broken though.
Oh.
You can see what happens. You have to be very careful with the really tiny things. I'm gonna look around for something else to pick up.
Yeah.
That's not broken. I'm not sure what that was anyway. Sorry, I'm just looking around a bit. Ismi are nice because they're nice and big. The rest of these ones are pretty small. But I do have 3 other ones I could look through. Uno momento. Oh, it's a cookie stack. I found a stack of cookies. You know how I feel about cookies. We're gonna get it. Coming for you, cookie. Oh. Did you see that? It jumped.
Yeah.
This cookie stack is playing hard to get.
Yeah. No.
Oh, there's also another isthmia right there. See if I can pick it up without doing the...
Oh, you left that one.
I did leave the cookie, but... I'm trying to get it up off of the... It's a little too close to the actual stub. Did it stay on there though? I think maybe it fell off.
You left it.
It fell off. It escaped.
We need to, I think, clean the drum.
Oh, you want me to clean off the needle?
Yeah. Okay, someone is asking what equipment do you use?
What's that?
What kind of equipment is it?
Oh, it's the name of the actual like maker? Is that what they're asking me?
I don't know, like, he's asking only what equipment do you use?
This Micro Manipulator is the MN151. The company is Narishi, Narisshige, and that's who made the Micro Manipulator. If that helps.
I hope so.
I got greedy and I lost it all. Including my cookie stack. The vertical direction is actually the hardest one to get a sense of because I can't very easily see how tall I am. Now I just need to pull this up. And there's our cookie stack. Easy as that, I guess. I was a little more successful when I had less junk on the tip of it. I even stacked it up for us. So ideally then you would get rid of all this debris, but you could basically just put little diatoms on your stub, and then if you had a really fine controller, you could probably reorient them, rearrange them. It requires a lot of patience, of course, which I may or may not have under any given day, but at the very least we can definitely put them in the position that we want. So there's that stack right there. You can see it kind of standing up on us, and adjacent to it is our isthmia. Right there. Pretty nice. Okay. Enough playing around with the Micro Manipulator. Some people demanding that I play tricks for them. I think that's everything I wanted to show people for today. So how close are we to 3? Hey, right after 3. Perfect. So I think that's going to be most of my stream. If you're interested in seeing some more, I'll probably play around with the Micro Manipulator again after we get some of the Gonfo monsters next week. Maybe on stream, maybe not, but we're probably going to rearrange some for scanning electron microscope analysis for real science, not just playing around with stuff. But you know, in our spare time, maybe we'll write out some messages to people using the Micro Manipulator. I'll get Rihanna to do that for me, so she can get some practice rearranging things and reorienting things without crushing them. Okay. Yeah, that's a sewing needle on the end of there. So I want to say thank you everybody for joining us today. It's been sort of a really different but fun stream. On Tuesday, I'm going to be interviewing Dr. Rachel Lupian. She's going to come on and talk about how she identifies paleoclimate change using leaf wax and finding leaf wax in lake sediments. And so it'll be pretty cool. She's got a really fun personality, and I'm just going to interview her. There won't be any microscopes or anything. That'll be at 8 o'clock on Tuesday evening. If you are jonesing for a fix on the microscope, tomorrow morning there should be a stream
Good news,
by Del
everyone.
Maximum on his light microscope for a bit. He does a nice casual morning stream, and on Monday night, Pacific Plankton will hopefully be able to do a stream looking at some of these same species that we just saw on these SEM stubs, but living material from San Francisco Bay, looking at plankton. So those two sciencey streams will hopefully keep you entertained between now and then. And then on Wednesday, we will probably be back on the scanning electron microscope doing a stream looking at some more of the material from Idaho with the Gonfo monster and this thing that we're trying to identify or describe. Yep, thanks everybody for hanging out with us, and I don't have any idea if there's anybody out there that we should raid. It's been a really fun stream doing something really different for us. So let's see if I can find somebody on my phone, or if somebody could make a suggestion, that's also fine. For people that I follow that are on. So Dr. WD-40 is an option. Oozebrood's an option. Boring Math Professor is an option. You know what? Deep Sea Jess looks like she's on. So why don't we go raid Deep Sea Jess? That'd be a good transition for us. So I'm going to set that raid up. Raid. Oops, I pushed the wrong button. My camera's in front of everything over on this thing. I did it again. think that's right. Looks like it is. So we'll go give Deep Sea Jess a raid. I haven't seen her for a while streaming, so it'll probably be nice for her to get a crowd of people coming in. And I want to thank everybody for hanging out with me today, doing something sort of new and fun, looking at things a little bit differently. We'll try to do some more frequent stereo microscope streaming now that I have everything set up in here and can kind of work with it a bit. So that'll be entertaining. Okay, everybody, I want to say goodbye now, and we'll catch you next time. All right. And also, I want to say thanks to Sukanya back there, who's been handling the stream for us throughout the whole stream, answering and helping sort of provide information about what chat has been saying. Okay, here we go.